Today we will discuss about a new paper where they have identified or where they are trying to find out the changes that occurs in the pancreatic cancer cells when they co-culture with this M2 macrophages. So in many of my previous videos we have discussed that M2 macrophages has the pro-tumorigenic properties. So however, what are the changes that M2 macrophages does in the PANC-1 cells that helps the PANC-1 cells in the progressions and metastasis, migrations, progressions and metastasis. So that changes are being targeted or find out in this paper. So we will discuss the first figure where what they have done is they culture the PANC-1 cells alone, and in the other case they culture the PANC-1 cells with the M2 macrophages, and thereafter they did this mRNA analysis at different time intervals. So and NPC is the control group where the only the PANC-1 cells are co-cultured, whereas NPM is the uh, targeted studies where PANC-1 is co-cultured with M2 macrophages or tumor-associated macrophages. So first thing that they did is they had to check the RNA expressions data. So RNA expressions data are RNA expressions in both the categories are almost equally visibles that can be seen with the diagonal plot, and same with the box plot we can see that RNA expressions and data are also in line with the are also similar to each other, that can be seen here in the box plot. And when they check the differentially regulated gene through the volcano plot, what they have find out is that in total 498 genes are dysregulated in the NPM or in the PANC-1 co-culture with M2 macrophages compared to the PANC-1 that is co-culture PANC-1 that is cultured without any macrophages. And out of these 498 genes that are dysregulated, 282 genes are upregulated, 216 genes are upregulated in the PANC-1 cell, and 282 genes are downregulated in these co-cultured PANC-1 cells. And when they check the differential expressions using the heat map, they can see that mRNA expressions are different when the PANC-1 cell is cultured alone compared to the PANC-1 cell is cultured with the M2 macrophage. And when they check the top 30 genes that are upregulated and dysregulated, they can see that genes that are upregulated are genes that are there are genes that are upregulated in the PANC-1 cells, and there are some genes that are upregulated in the uh, for the panel E. Let me restate what this panel E represents. Panel E represents the top 30 genes that are upregulated in the PANC-1, which is co-cultured with the M2 macrophage or NPM group compared to the NPC group. And there are 30 genes that are top 30 genes that are downregulated in the NPM group compared to the NPC group. That is shown in this panel E, where red color represents the upregulations. So these are the upregulated genes in the NPM group, and these are the upregulated genes in the or these are the downregulated genes in the NPM group. And after that, they check the top genes that are top genes that are highly upregulated. So what they have found out is that the VCAM seems to be highly upregulated. It has the 7.06 fold chain with a p-value of less than 3.26 into 10 raised to minus 5 and FDR value of 0.04. So this becomes their fundamentals of to check the effect of VCAM on this PANC-1 co-cultured cells or in the NPM group. So In this panel E, we can see that VCAM is not at the top space, but when we combine these three conditions, that is fold change, FDR, and p-value, this VCAM comes at the top place, is being highly upregulated. So that's all for this video. So in this video, they provided just a characterization of the mRNA expressions in the NPM group and NPC group, and they have find out that VCAMs is the highly upregulated group, and they will do their other experiments based on this informations. If you need to understand more about their experimental procedures or their bioinformatic approaches, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.